Okay. All right. We left off yesterday going over the synthesis of purines, nucleotides, and I just kind of want to review just a little bit on the synthesis of purines. So remember that in the first step we start with PRPP. So PRPP is going to serve as the starting material. Hold on, I just want to make sure I actually started recording. Oh, yes, I did. Okay. So PRPP is going to serve as the starting material. PRPP is going to provide that ribose backbone for assembling the base upon the ribose backbone. And it goes through all of these different steps that involve adding different carbons and nitrogens and hydrogens and oxygens. And they're coming from really intermediates of metabolism or from amino acids or different cofactors. And it leads to formation of this IMP intermediate. And then IMP can be further modified to make AMP and GMP. And again, for understanding sort of what you need to know in terms of the structure, you should just know where each atom of the purine is coming from. And so purine synthesis is pretty well regulated. So there's essentially product feedback inhibition, and this occurs really at three main steps. That is the first and second step. So the first step in which ribose 5-phosphate is made into PRPP, that's a point of regulation, as well as the actual first step of purine synthesis, which is the conversion of PRPP into 5-phosphoribosylamine. That's a regulatory step. Another regulatory step is sort of that branch point at which IMP is converted into AMP and GMP. So if there's high levels of AMP, then AMP is going to feedback and inhibit the enzyme that's going to lead to the formation of more AMP. So that makes sense, because if there's lots of AMP in the cell, then we don't need to make more of it. Similarly for GMP, if there's high levels of GMP, it's going to feedback and inhibit IMP dehydrogenase. Another thing is both AMP, GMP, as well as IMP, if there's high levels of that, that's going to go back and feed and inhibit glutamine, PRPP, aminotransferase. This is, again, similar idea that there's high levels of products. We don't need to continue making more of the products. And then ADP can also act as an inhibitor of this pathway. Okay, so here's sort of what you need to know for purine synthesis. You need to know how PRPP is made and the role of PRP. P meaning that it's going to involve as sort of the backbone for synthesizing the base. You need to know the first two reactions of the synthetic pathway and how it's regulated by AMP, GMP, and IMP, where the atoms in each base come from, and that IMP is going to be a common intermediate to make AMP and GMP. Okay, so you don't need to know every single reaction for the purine biosynthetic pathway. You just kind of need to know sort of this general information. So I'll give you guys a chance to write this down or take a picture of it. And I have it recorded in my lecture as well if you need to refer back to it. Grab a sip of water. Okay, so we're going to very lightly cover pyrimidine synthesis. Now, pyrimidine synthesis differs from purine synthesis in that you actually make the 6-membered pyrimidine ring first, and then you attach it to the ribose backbone. So you attach it to ribose 5-phosphate. Now, pyrimidine synthesis actually involves carbamyl phosphate. This is synthesized by carbamyl phosphate synthetase 2. And this is actually the cytoplasmic enzyme. So this is actually different from the mitochondrial enzyme that catalyzes the same reaction. So the mitochondrial enzyme is CPS1. Where have we heard of CPS1 before? In the urea cycle, right? So CPS1 makes carbamyl phosphate, which carbamyl phosphate goes into the urea cycle. And so CPS2, which is in pyrimidine synthesis, it catalyzes the same reaction, and it does so by the same exact mechanism where it takes ATP, or it binds ATP and bicarbonate. It reacts bicarbonate with ATP and phosphorylates it, making a carbonic phosphoric, or carboxy phosphate intermediate. An amino group can then attack the carboxyl group and displaces the phosphate, making carbamate. And then addition of another phosphate group coming from ATP makes carbamyl phosphate. So this is just a review of a reaction mechanism we've already learned. Pat?
In the urea cycle,
Yeah.
carbamyl phosphate, is it synthesized in the mitochondria?
Yes, it's a mitochondria enzyme.
This is physically distinct.
Yep. So there's CPS1 that's in the mitochondria, and that's specifically for making, carbamyl phosphate to go in the urea cycle, and then CPS2, which is the cytoplasmic form, which is involved in pyrimidine synthesis. Great question. Okay, so in pyrimidine synthesis, the first reaction that occurs is that's catalyzed by this enzyme called aspartate transcarbamylase. So you guys will have to learn this enzyme, but aspartate transcarbamylase will take ATP and react it with carbamyl phosphate, and it'll transfer the carboxyl group onto aspartate, making N-carbamyl aspartate. Okay, so that's the first reaction in pyrimidine synthesis. Really, pretty much the only reaction you have to know. Okay, and then later reactions involve, there's later modifications followed by ring closure until you take the 6-membered ring, and then you react it with PRPP in order to then attach that 6-membered ring onto that ribose 5-phosphate backbone. So this differs from purine synthesis. In purine synthesis, you actually started with PRPP, that ribose backbone, and you made the base using that backbone as a carrier. In pyrimidine synthesis, you make the base first and then attach it to the ribose 5-phosphate backbone. So that's, I would say, a pretty important thing to know. Okay, and then once you make this intermediate through other reactions, you make UMP, and then through another reaction, you make CTP. So CTP is made from UTP. Now, this is what you need to know is that CTP is going to be one of the end products of the pyrimidine biosynthetic pathway, and this product is actually going to feedback and inhibit the first step of the reaction. Okay, so if there's too much or there's plenty of CTP around, then we want to shut off this pathway, and we always do that by inhibiting the first enzyme of that pathway, which is to inhibit aspartate transcarbamylase. So CTP acts as an allosteric inhibitor of the enzyme. And so that is one of the mechanisms, or a major mechanism, by which pyrimidine biosynthesis is regulated, is through the regulation of aspartate transcarbamylase. That first step is the committed step in pyrimidine biosynthesis. This is the structure of aspartate transcarbamylase. It contains regulatory subunits, so those are the red and yellow subunits, and then the catalytic subunits are these blue-colored subunits. Both CTP and ATP can bind to the regulatory subunit and causes a conformational change, which then affects the activity of the enzyme. And so CTP, which is the end product of the sequence, is going to inhibit the enzyme, so it's going to essentially slow down the reaction. And then ATP actually activates the enzyme. And so one of the rationales why ATP actually activates the enzyme is if you have lots of ATP around, that means you have lots of purines around. So we need to sort of compensate for greater concentrations of purines by making more pyrimidines, okay? And that's why ATP will activate aspartate transcarbamylase, which will then increase pyrimidine synthesis. So that way you have sort of equal amounts of purines and pyrimidines around. Okay, so here's what to know about pyrimidine synthesis. CPS2 and the production of, should be carbamyl phosphate. The aspartate transcarbamylase reaction and how it's regulated, and the fact that PRP is going to react with the ribose ring and serve as that the ribose 5-phosphate backbone. So you had a 6-membered ring that's synthesized first, and then you attach it to the ribose of PRPP. And that's about all I really need you guys to know. So I'm sorry that you guys don't get to learn all the reactions, but you certainly can on your own if you want to. So here's just some sort of common themes. We can see that PRPP is actually important for both purine and pyrimidine biosynthesis, although they're kind of coming in the biosynthetic pathways at different times. We can see that amino acids they serve as important precursors for each pathway. So glycines kind of sort of start the purine pathway, and aspartate start the pyrimidine pathway. We can see that glutamine is actually a very important source of amino groups in 4 different steps of the de novo pathway. Aspartate is also used as a source of the amino group in the purine pathway. Now, the cellular pools of nucleotides are actually quite small. So 1%, it's actually 1% or less than the amounts that are required to synthesize the cell's DNA. And so the cells have to continue synthesizing nucleotides during nucleic acid synthesis or replication. And in some cases, the actual pool of nucleotides as well as the rate of nucleotides synthesis can actually limit the rate of DNA replication and transcription. And so this is why often the nucleotide synthesis pathway is actually an attractive target for antibiotics and chemotherapy agents. So many chemotherapeutic agents target these nucleotide synthetic pathways. So cancer cells, remember, a cancer cell is defined by a cell that grows and divides uncontrollably, so it's doing lots of replication. They have a greater requirement of nucleotides as precursors of DNA and RNA, and they're going to be more sensitive to inhibitors, to nucleotide biosynthesis inhibitors versus normal cells. So normal cells, they generally divide very little. They replicate and divide very little, or not at all. Whereas cancer cells, they're replicating and dividing all the time, so they're much more sensitive to these inhibitors. One important class of chemotherapeutic agents that that target these enzymes in these biosynthetic pathways are these glutamine aminotransferase inhibitors. So the glutamine aminotransferases, they're in these pathways. They're important for, transferring the amino groups from glutamine onto these onto the nucleotide bases. And so these inhibitors, they actually look very similar to glutamine. So an example is azacitidine, and I don't really know how to say it, acavin. They look very similar to glutamine, and they actually bind in the same site that glutamine binds in these biosynthetic enzymes. And so these biosynthetic enzymes, they're really strongly inhibited by these inhibitors, and that is used as sort of a cancer inhibitor or a chemotherapy. Okay, so now we're going to talk about reduction of ribonucleotides to deoxynucleotides. So ribonucleotides are actually the precursors of deoxyribonucleotides, and it's catalyzed by the enzyme called ribonucleotide reductase. So what it does is it reduces the 2' position of the ribose ring of the ribonucleotide, and this involves NADPH as sort of the ultimate donor of electrons. So the hydroxyl group is reduced and replaced with a hydrogen at that 2' carbon. And the reaction is really quite unusual because it involves this carbon that's not activated and a free radical enzyme mechanism. And then once deoxynucleotide diphosphates are made, then a separate reaction is going to convert the DNDPs into DNTPs. And so, we're really going to skip this slide, but I'll just talk about this very briefly. This is the structure of ribonucleotide reductase. It has regulatory subunits and catalytic subunits, and essentially different nucleotides can bind to the regulatory subunit, which then actually dictates the specificity of the catalytic subunits. But again, you guys don't have to know this information. We're going to go ahead and skip this information. And we're going to talk about how it is that deoxy TMP is made. So uracil is the closest base to thymine, and so DUMP is used to make DMP, so deoxythymine monophosphate. So this is catalyzed by the enzyme called thymidylate synthetase, and it essentially binds DUMP as well as a cofactor called N5N10 methylene tetrahydrofolate. And it does a 1-carbon transfer. So it transfers a methyl group from methylene tetrahydrofolate onto the base structure of DUMP to then make DTMP. And then this also produces 7,8-dihydrofolate. Now, this is a cofactor or coenzyme, and it has to be regenerated in order to be able to keep this thymidylate synthase reaction going. And so this involves the enzyme dihydrofolate reductase, which reduces 7,8-dihydrofolate into tetrahydrofolate, and then another transamination reaction, which then converts tetrahydrofolate back to the N5N10 methylene tetrahydrofolate, which then can be reused for the thymidylate synthase reaction. And so both this enzyme, thymidylate synthase, as well as dihydrofolate reductase, these are actually targeted in cancer treatment. And so there are inhibitors that actually specifically target these enzymes, and one of these is 5-fluorouracil. So this is the structure of 5-fluorouracil. It's been a major anti-cancer drug since the 1950s. In the body, so it comes as a drug, 5-fluorouracil, and then we take the drug, and then the 5-fluorouracil is actually metabolized into fluorouracil DUMP. So this is the structure, well, here, this is the structure of the fluorouracil DUMP. So it's essentially been attached to a ribose monophosphate. And this is sort of the mechanism by which it works. So this is sort of the actual mechanism of thymidylate synthase, by which it takes DUMP and transfers a methyl group onto there. The enzyme has the cysteine in its active site. It binds DUMP, and then the cysteine, the sulfhydryl group of the cysteine, attacks the carbon on DMP, and then places the electrons onto an adjacent carbon, resulting in this carbanion nucleophile. The carbanion then nucleophilically attacks the, that methyl group on the N5-N10 methyl hydrofolate and forms this covalent adduct. And then you have this hydride shift. So you have a hydride shift of this hydrogen to this carbon. Which then transfers the methyl group onto the substrate, and then it breaks this carbon-carbon bond. So then that releases the substrate from the enzyme. So the way that this drug works is that the fluoro-DMP, it'll actually react with the enzyme in a similar mechanism. So the cysteine, the sulfhydryl group of the cysteine will attack the carbon, resulting in the carbanion intermediate. It'll then attack the methyl group of N5-N10 methyl tetrahydrofolate, and then essentially it gets stuck. It forms this covalent adduct, which cannot become reversed. So it forms this dead-end covalent complex, which essentially kills the enzyme and can no longer catalyze another reaction. So that's how this drug works in terms of acting as an anti-cancer agent to essentially inhibit the synthesis of DUMP, and then that actually inhibits, right, DNA replication, because you need thymine nucleotides to be able to replicate DNA. Okay, so the 5-fluorouracil specifically inhibits thymidylate synthase. In addition, dihydrofolate reductase is also a major target of cancer agents. Okay, so these drugs called antifolates, they are also chemotherapies. So antifolates are drugs that basically inhibit this pathway that essentially recycles the N5-N10 methylene folate. So one of these, so the way that they actually work is they actually inhibit this enzyme, dihydrofolate reductase, which converts 7,8-dihydrofolate into tetrahydrofolate. And so one of these is called methotrexate. And methotrexate looks very similar to 7,8-dihydrofolate. So it acts as kind of like a competitive inhibitor. So it inhibits dihydrofolate reductase, and that means that the 7,8-dihydrofolate cannot be recycled into the N5-N10 methylene tetrahydrofolate, which then this reaction can't occur. So since folate is essential for the synthesis of nucleotides and some amino acids, then blocking it has the most effect on rapidly dividing cells. So that's going to be cancer cells, but it also is going to affect other type of rapidly dividing cells as well as bone marrow, hair, cells that are involved in the gastrointestinal system. So that also tends to be why cancer patients, when they're taking these type of drugs, they tend to lose their hair, or they get very upset stomach, and that's because these drugs are anti-cancer drugs. While they're targeting the cancer cells, they're also targeting the rapidly dividing cells as well. Okay, I'm going to skip this slide. You guys can read it on your own, but you won't be responsible for it. And let's see. Okay, so we're going to skip the catabolism of pyrimidine nucleotides. And the only thing I wanted to talk about was very quickly is the catabolism of purines. So purines can be, they need to be catabolized and broken down. So essentially GMP and AMP, they are hydrolyzed to essentially the free bases. And then the free bases are then oxidized to uric acid. So this is the structure of uric acid. Now, uric acid may actually play protective roles in the body as an antioxidant. However, excess uric acid actually has a pathological role. So if there's an excess of it, it can accumulate in the hands and feet, and it can actually form crystals, which can lead to gout. So gout is a very, very painful disease, and it seems to be more prevalent in men than women, or it is more prevalent in men and women. And so one of the drugs that people use to treat gout is this drug called allopurinol. So this is a drug that inhibits the enzyme that converts hypoxanthine to xanthine in the pathway, as well as avoiding a very rich diet. So diets that have very nucleic acid foods are also thought to contribute to acquiring gout. Okay, so I'm going to have you guys skip the salvage pathways, and that concludes nucleotide metabolism part 2. So do you guys have any questions for nucleotide metabolism?
Could you go over what slides you skipped and we don't need to know? The antifolate and antibacterial salvage pathways.
Yeah, so let's see, what do you guys not need to know? Okay. So you guys don't need to know this slide. Okay. And so this is the slide on ribonucleotide reductase. I mean, you still have to know that ribonucleotide reductase is the enzyme that converts ribonucleotides into deoxyribonucleotides, but you don't have to know the specific information. You don't have to know this slide, although it's quite interesting because antifolates also act as antibacterials. That's because bacteria grow fast as well as, in addition to cancer cells. You don't have to know the catabolism of pyrimidines. And honestly, the only thing that I want you guys to know in terms of the catabolism of purines is how it results in gout. So that's what I want you guys to know about that. And then we don't have to know about the salvage pathways. Anything else? All right. I'm kind of happy to finally leave the nucleotide metabolism chapter and move on to genes and chromosomes. We get to finally talk about one of my favorite topics, which is epigenetics, and that's going to come later in the talk. Okay, so now we're going to go into genes and chromosomes. So what is a gene? Gene is the fundamental unit of information in living systems. It's defined biochemically as a segment of DNA, or in some viruses, RNA, that encodes the information required to produce some type of functional biological product. And that final product is usually a protein, but it can also be an RNA molecule. The genome is all the genetic information that is encoded in a cell or virus. DNA in a eukaryote or prokaryote, it's organized into chromosomes, and the chromosome organization is important for organizing the gene, and that chromosome will contain thousands of genes as well as other DNA tracks. So there's other DNA content in the chromosome. Prokaryotes, they have a single circular chromosome, and then eukaryotes, they have multiple linear chromosomes. There are 3 major processes that the cell uses for using the genetic information. This is essentially called centrodogma, which you probably heard from your biology 120 or your genetics class. And so replication is the process by which the DNA of a parental strand is copied into daughter DNA molecules that are identical in sequence. Transcription is the process by which parts of the DNA message is copied into RNA, and then translation is the process by which RNA, that message that's encoded in RNA, is translated into protein by ribosomes. We're going to skip this slide and go on to gene expression. So gene expression is collinear. So collinearity means that the coding nucleotide sequence of DNA and mRNA corresponds to the amino acid sequence of a polypeptide chain. And so if we have a double-stranded, a strand of double-stranded DNA, one of the strands of DNA is going to serve as the template for synthesizing the mRNA strand. And then the mRNA strand has these nucleotide triplets, so these codons of 3, repeating units of 3, which are, they're going to be complementary to the DNA. And those codons, they are going to essentially code for an amino acid. So 3 nucleotides codes for 1 amino acid. Now, in some bacteria and many eukaryotic genes, the coding sequences, or exons, they're interrupted by non-coding sequences, or introns. So exons specifically are actually collinear and introns are not. Okay, so DNA molecules, they are very, very long, much longer than the cellular packages that actually contain the DNA. So let's consider E. coli, which is kind of our sort of the model organism for a unicellular prokaryotic organism. The E. coli is well studied, and we have sequenced the genome. So the E. coli chromosome, it's just one single double-stranded circular DNA molecule, which is located in the nucleoid region of E. coli. So remember, the nucleoid region is really kind of the region of E. coli that's just kind of dense with DNA, or enriched with DNA. It contains about 4.6 million bases, or base pairs, that, if you were to stretch it out, has a length of 1.7 millimeters, and that's about 850 times the length of the actual diameter of an E. coli cell, which is 2 microns. So it's a lot of DNA fitting into a very small E. coli cell. And then the base pairs of the genome of E. coli encode for about 4,300 genes, which are protein coding, and then about 115 genes that code for these stable RNA molecules. In addition to the one circular chromosome, many bacteria also contain plasmids. So plasmids are these small circular DNA molecules. They're also double-stranded, and they're kind of found freely floating around in the cytosol. And they contain sort of these extra chromosomal elements that, in most cases, doesn't help the bacteria. The plasmid tend to be either a few thousand base pairs long, but some are even larger, so they can be greater than 10,000 base pairs. And they can also carry genetic information, where that genetic information can also encode for protein. When E. coli and bacteria undergo replication undergo replication, they also replicate the plasmid as well. So the plasmid also will become replicated, and then those replicated plasmids will then be passed into the daughter cells at cell division. And plasmids are also found in yeast as well as other fungi, in addition to bacteria. Plasmids are kind of considered parasitic. However, there are some cases where there is a mutualistic interaction between the plasmid and the host. So the plasmid might carry genes that actually help the host, for instance, carrying a drug resistance gene. So So, you know, it could be that the E. coli might acquire a plasmid that allows it to become resistant to different types of antibiotics. Okay, so this table shows essentially it kind of compares the total amount of DNA as well as the number of chromosomes and number of genes for these different types of organisms. If we kind of look at the simpler organisms, such as yeast and bacteria, we kind of see this trend in that these simpler organisms, they have fewer genes and sort of less DNA. However, as the complexity of organisms sort of increase, so we have worms, plants, fruit flies, another type of plant, mice, and humans, there is an increased number of genes, but that increased number of genes kind of levels out around somewhere around 30,000 to 50,000. And instead what we kind of see is that the total number of base pair actually increase as the complexity of the organism increases, suggesting that the DNA has some type of, it has a regulatory function for regulating the genes. So let's talk about eukaryotic chromosomes. Eukaryotic chromosomes, the genetic material of the eukaryotic cell is divided into chromosomes. Each chromosome is a single, very large duplex. So a duplex is a double-stranded DNA molecule. And then the DNA molecule in the 24 different types of human chromosomes. So there's 22 matching pairs plus the X and Y sex chromosomes. They vary in length. And then each chromosome carries a sort of a unique set of genes. So eukaryotic chromosomes is essentially the way that it allows the cell to be able to compact the DNA into an extraordinary degree. So if you were to place the whole human genome, so you were to stretch out the DNA of each of the 22 plus the X and Y chromosomes out end to end, that DNA in just one cell is approximately 1 meter long. And if you consider the fact that most cells are diploid, then each cell contains about 2 meters of DNA. And since each human body contains about 10 to the 14 cells, then that's a total of DNA that's about 2 to the 11th kilometers in length. And if we compare that to like the circumference of the Earth, which is 4 times 10 to the 4th kilometers, or the distance of the Earth to the Sun, that's 1.5 times 10 to the 8th kilometers, then you can see that, right, our bodies have a ton of DNA, and it's able to really be able to compact that DNA into the nucleus of the cell, which is quite amazing. We're going to skip this slide about mitochondria DNA, and then just kind of talk about sort of the difference between eukaryotes and prokaryotic genes and chromosomes. So remember that many bacteria species, they have only 1 chromosome per cell. In nearly each case, each chromosome contains only 1 copy of each gene. So very few genes are repeated several times. In prokaryotes, almost every gene is precisely collinear with the amino acid sequence or RNA sequence. In eukaryotes, the nucleotide sequence, some of it is collinear, and that's really going to be the coding sequence, which are the exons, and that's broken up or intervened by DNA that does not encode for amino acid sequence. So those are going to be the introns. Okay, and so eukaryotic genes and chromosomes are very complex. So the intervening sequences, those are introns. Those are going to be the DNA segments and the genes that are not translated. And exons are going to be the coding segments. So those are going to be the parts of the gene that will be coded into protein. And so few prokaryotic genes actually contain introns, whereas in higher eukaryotes, the gene sequence of a eukaryote actually contains more introns than exons. And why exactly there are introns isn't entirely clear, although, well, it is kind of clear in some cases, but at least not all the functions of the introns are not clear. Okay, so in terms of type of sequences in the human genome, let's just consider genes. Okay, so if we just look at the exons, so those are that's the part of the human genome that's going to be encoded into protein. That's only about 1.5% of the human genome, which actually carries information for making protein or RNA products. If you then sort of include the larger intronic portion, then that counts for about 30% of the human genome. And so this leaves a lot of other DNA, so about 70% of the human genome that is not information encoded in genes. And so this leaves a lot of other DNA that has other roles. So one of the types of DNA are these DNA elements called transposons. So they're segments of DNA that range from just a few hundred to several 100,000 base pairs long. Transposons, they are able to move from one location of the genome into another location. So that's why they're called transposons, because they can kind of transfer from place to place in the genome. So they're sort of a molecular parasite, and they are able to sort of make a home within the host genome. These transposons, transposon gene areas, they have genes that encode for protein that actually catalyze the transposition process. So it actually catalyzes proteins that allows the segments to be able to move from one area to another in the genome. Some transposons are quite active, so they can move at a low frequency, but most are actually inactive, where they're kind of sort of forced to stay where they are. So they're inactivated and altered by mutations. They generally do not encode proteins or RNAs that are actually used by human cells, and they have played a role in human evolution. So there's been major movements of transposons, and it can lead to the redistribution of other genomic sequences. There's essentially 4 classes of transposons. There's the long interspersed elements or lines. They are 6,000 to about 8,000 base pairs long. And they typically include the few genes that catalyze the proteins that catalyze transposition. There are the short interspersed elements, so SINEs, so those are about 100 to 300 base pair longs. And then there's the retrovirus-like transposons. Those are about 1,500 to 11,000 base pair longs. They're actually trapped in the genome, so they're kind of like stuck there, so they can't move from one to another. And they're actually related to retroviruses, such as HIV. So they're actually coming from viruses, other ancient viruses. Okay, and then there's other miscellaneous sequences. So there's these SSR, so simple sequence DNA repeats, or highly repetitive sequences. These are very short sequences, less than 10 base pairs in length, and they're repeated sometimes up to 1 million times per cell. Yes, Mitch?
Sorry, so there were 4, but then there were 3. Is the SSRs a transposon?
No. So this is sort of like the miscellaneous. So now we're talking about SSR, and now these are sort of the miscellaneous.
Okay.
Yes.
So are there 3 transposon classes then?
Yeah, it's actually 3, not 4. Apologize for that. Okay, so then there's SSRs. So those are kind of these repetitive sequences. They don't encode for proteins or RNAs, and they have actually a functional importance. So they're associated with essentially centromeres and telomeres. And then there's also large segmental duplications. So essentially these are, it's literally what it sounds like. These are essentially large segments of DNA that are duplicated, and they appear in more than one different location. So you can have a duplication of DNA in one chromosome, and then a duplication of DNA in another chromosome. And then there's also what we call these unlisted elements, where we don't really know exactly what they do. But going back to the SSR sequences, again, they're found in the centromeres and telomeres. So a telomere is a sequence of DNA that is important for cell division as an attachment point for proteins that link the chromosome to the mitotic spindle. And it's important for this equal and orderly distribution of the chromosome sets from sets to the daughter cells. And then there's the telomere sequences. So these are going to be the sequences at the end of the chromosome that help to stabilize the chromosomes. So the telomere repeats, the telomeres have these repeats. They range from about 20 to 100 base pairs long in single-cell eukaryotes, and then about 15,000 base pairs in mammals. And so these telomere repeats actually have a very important role, and that's because the end of a DNA molecule, they can't be they cannot be replicated by cellular replication machinery. And so the enzyme called telomerase actually adds these repetitive sequences to the end of the chromosomes. And that helps to essentially stabilize the chromosomes. Okay. All right. So I'm going to just talk for like 5 more minutes, and then we'll cut the lecture early, okay? Just because it's like been a very lecture-heavy class session, and then I'll give out the quizzes. So let's talk about DNA. We're finally going to get into the material that, to me, is very interesting to me, which is this tertiary structure of DNA. So let's talk about DNA supercoiling, which is the basis of tertiary structure. Now, supercoiling means the coiling of a coil, okay? Now, DNA is a helix, right? So that's a coil. And if you were to actually further coil the axis upon itself, so actually kind of, it's a lot like if you were to take a telephone cord. So I know no one owns like telephone cord. Okay, so you guys have seen them, right? Telephone cords.
My grandparents still have them.
Well, there you go. If you were to take a telephone cord, right? The telephone cord itself is a coil. If you were to stretch it out, okay, and then relax it, what happens? It coils upon itself, and then you get this big, ugly, dangly coiled mess, right? That's what our phone used to look like when we were growing up. In fact, it probably still looks like that. Okay, so that's what supercoiling actually is that you have the coiling of a coil. So that's what DNA does. Is it's a coil structure, and then if you were to sort of induce some type of strain on it, it'll cause it to further coil, and that induces supercoiling. And that supercoiling is really important because it allows it to sort of fit into a smaller space. And we just talked about the fact that there's lots and lots of DNA in a eukaryotic or prokaryotic organism relative to the space it has to fit into the cell. So it's going to be very important for packaging. The other thing about supercoiled DNA is that it actually helps the DNA move faster through a gel during electrophoresis. So if you were to it's called a DNA gel agarose electrophoresis to essentially separate the different pieces of DNA. So if you were to take a supercoiled piece of DNA versus a non-supercoiled DNA, it'll move a lot faster because it's smaller. Okay. Now, when there is no net bending of the DNA axis upon itself, it's said to be in a completely relaxed state. So this is a circular DNA plasmid where there's no supercoiling at all. It's an electron micrograph of the plasmid. It's in a completely relaxed state because there's no supercoiling that occurs. However, if you were to increase supercoiling or increase strain on the DNA, that will then increase the supercoiling. So these are different images of that same DNA plasmid that has more and more supercoiling. You can see that it kind of further and further compacts the DNA. Now, it's the structural strain that actually induces supercoiling, and specifically, it's the underwinding of the DNA that induces it. So, well, in most cases, underwinding of the DNA. You can have underwinding and overwinding, but usually it's underwinding that actually supercoils the DNA. Underwinding is when you actually have fewer turns in the helical DNA molecule as expected. So for example, if you have a double-stranded piece of DNA and it's completely relaxed, that piece of DNA may have 8 turns, okay, in that DNA. If you were to then underwind it, so that way it has instead 7 turns, and strain it, then that's going to then cause it to form a supercoil. And so the cell actually maintains DNA in kind of this underwound state that facilitates compaction and supercoiling, as well as the act of separating the 2 DNA strands, so pulling the 2 pieces apart from each other, that actually causes strain and causes supercoiling. And strand separation occurs a lot because it's associated with different DNA processes. Okay, so enzymes called topoisomerases, they are enzymes that can increase or winding of DNA by essentially hydrolyzing the phosphodiester bond of either one of the strands of the DNA strand or both strands of the DNA. So the type 1 topoisomerases, they actually hydrolyze the phosphodiester linkage in one strand of DNA to either induce or relax the supercoiling. And so what they do is the enzyme binds the double-stranded piece of DNA to one of the strands, a tyrosine, an active site tyrosine will actually attack the phosphate group in the phosphate backbone of DNA, which then breaks the bond between the phosphate and the oxygen that is attached to the 3' carbon of the DNA backbone. So it essentially causes a break in one strand of the double-stranded DNA. Okay, and then at this point, this results in a topoisomerase DNA adduct. And then what happens is the enzyme will actually change conformation, and it'll actually take the unbroken DNA strand and kind of pass it through the broken DNA strand, and then it'll actually reverse this reaction. So it'll catalyze a reaction in which the hydroxyl group of the 3' carbon of the phosphate then reacts with the phosphate of the enzyme, the covalent enzyme intermediate, which then releases the reactive tyrosine, remaking the phosphodiester bond. Type 2 topoisomerase, they essentially do the same thing, except they hydrolyze both phosphodiester linkages to either relax or induce a supercoiling and then reseal the DNA. All right, so we're going to stop it there.